Oh, yeah. There you go. All right. And I think it's recording. Looks like a little blinky red light.
That's good. Yes.
Go team.
Go team. Go. Do that before I make some choice.
It's just whack. This professor's just no good.
Good afternoon, everyone, and welcome to our first Hawk Talk of this semester. Thank you all for joining us today. Today we're going to have Dr. Nathan Green discuss his research on engineered nanoparticles. He's going to focus on recent efforts at NSU to design, functionalize, and assemble complex nanomaterials to explore novel solutions to the next generation of engineering challenges. So without further ado, please join me in welcoming Dr. Nathan Green.
Thank you very much. Thank you for the invitation and for the introduction. So my name is Nathan Green. I'm an assistant professor of chemistry here on the Broken Arrow campus at NSU. I'm in my third year now, so these are all some recent efforts. But I'd like to talk about some of the research that we've been doing in my lab, particularly highlighting the support we've received from the Faculty Research Council in order to do some of this work. So I've entitled this Small Particles, Big Impact, because I think it's funny and I enjoy that. But one of the things that I enjoy so much about nanomaterials research is taking very small objects and manipulating them to have a larger impact. So getting small things to behave in a particular way so you have a bulk property coming out of it. And I'll try and carry that theme through. There we go. So what we'll do first is go through a bit of background, and I'll try and convince you that nanoparticles are actually pretty cool and that you've seen them before. You maybe just haven't realized it. I want to talk about some of the research that I did that led up to why my lab is focused on engineering nanomaterials and what they could potentially be used for, and then get into some of the work that our fabulous undergraduate researchers have been getting into and highlight some of their accomplishments as we carry on here. So there are numerous applications for nanoparticles, but I want to highlight a couple that you maybe have seen before. I have actually not seen this cup. This is called the Lycurgus cup. It is a Roman artifact that when you have visible light shown on the outside of it, so if it's just sitting in this room, it would look green to us. But if we had light shining behind the glass, it looks red, kind of glows reddish color. For a long time, this cup is one of the only examples of this type of glass decoration in Roman architecture or in Roman artifacts. And we didn't know why. But a small piece of this cup broke off and it was studied. And under a scanning electron microscope, a transmission electron microscope, we found that there are several little particles that make up the cup. And it turns out that those little particles are gold nanoparticles. If that color, that kind of deep wine red color looks familiar to you, because you've also seen it in stained glass windows. In older cathedrals, especially throughout Europe, there are brilliant reds in most of the stained glass windows. And you notice this is just one example, but the color that you see from the blue and the yellow is not nearly as stark as that deep red. And you see that deep red color used a lot in stained glass windows. That comes again from imparting silicon nanoparticles into the matrix of the glass. The ancients didn't know what they were doing when they put this together, but they poured in a salt precursor that had gold salt in it and a reducing agent. And as the glass cured, so it's molten, and as it cured, gold nanoparticles got captured into the matrix there, and that left the glass looking this kind of deep red color. There's other ways to make stained glass as well, but one of the most famous examples is suspended gold nanoparticles. If they had known what they were doing, they could have made all of the different colors out of nanoparticles. And so this is taken from the Merkin lab, probably one of the most highly published nanomaterial scientists in the world right now, where he brings up examples of if you take gold and just simply make it different sizes, so as they get larger, they start to reflect different colors, and we'll talk about why that is, but you could make a whole variety of colors from nanoparticles, and it comes from their electronic properties. In addition to just being pretty and making decorative objects like Roman cups and stained glass, you also see gold nanoparticles used in diagnostic tests quite a bit. This is a particular type of test called a laminar flow assay. What happens is you put some sort of analyte, typically a solution of something you're trying to find. It's soaked into a pad, runs into another pad that has gold nanoparticles and antibodies attached to it, and that continues flowing down the pad through capillary action, where then our gold nanoparticles and antibodies can bind to any analyte that's in solution. They get carried across to where there's two strips with matching antibodies, and there will be one set that matches up that you should have gold nanoparticles gather as a control, and one set that the gold nanoparticles will only gather there if there's the correct protein. So in this case, what I'm getting at here is a pregnancy test. This is the main thing we would see these used for. The protein in question would be human chorionic gonadotropin. If HCG is present in the sample, it will bind across these antibodies, and you'll see a strip of gold nanoparticles here. This strip, and this is not a great cut, there should be another protein in here that causes those antibodies to match up. And that's where you see this classic example of the two-line test you see in pregnancy tests. One line shows that, yes, we're getting color where we should get color. The other line shows up if the particular protein of interest is present in the sample. And this is something I didn't know until actually a few weeks ago, but a lot of pregnancy tests have that red color to them. That's from the gold nanoparticles. It's the same red that you saw in the Lycurgus cup. It's the same red that you saw in stained glass. And so I kind of pulled up this image and blow it up a little bit more. This is from my lab of turning the precursor salt chloroauric acid into gold nanoparticles. That deep red color, you can see it's like the same thing. So these are the gold nanoparticles in solution, and those are the gold nanoparticles gathered on the pregnancy test. And so the question might be, why are we putting gold in pregnancy tests? It seems a little bit extreme, but you can imagine that someone wants the right answer when they take a pregnancy test. And so if they're using a colorimetric assay to figure out if HCG is present in their body or not, they really want to know if this second line, so this is the control line, they really want to know if this line is going to show up or not. So if you use something like a dye, say from like a marker, those photo bleach, or they can dissolve, or they can be oxidized in solution, and they can lose their color. We need that color to be preserved. So gold nanoparticles are quite stable, and they maintain their color. They're not going to photo bleach. If I left this pregnancy test in the sun, it's not going to fade away. The gold nanoparticles will gather in that place and hold out there. So why are they red? If anyone seen block gold before, you might notice that one of its main characteristics is it is not red. It is gold in color. So why are they red when you make them smaller? Well, it turns out chloroauric acid, that salt I was telling you about, is kind of a goldish color. So this is, if I was an alchemist, I'd be very interested in this. This is liquid gold. It's a liquid, it's a aqueous solution of gold-3. And when you put it into a reducing agent, namely a molecule called citrate, and you boil it, you can turn the gold-3 into gold-0. So you get gold atoms to start binding together, and they make up eventually a little marble. And that little marble is very, very small. So this is 50 nanometers. A wavelength of light, one of the shortest that we can see, is on the order of 300 nanometers. So we're a good deal smaller than a wavelength of light. You can't visualize these with optical microscopes. We have to use electron microscopes to see them because they're too small. But what happens, because they're smaller than a wavelength of light, it means that they are subjected to the electromagnetic radiation of a wavelength of light. So that's kind of illustrated up here. The particle itself is shown as that silver ball. And you might know that gold's a good conductor of electricity. It's because there's a lot of free electrons in gold, so we can move those electrons around. The electrons around the gold are shown as kind of the green ball here. So light is electromagnetic radiation. It is an electric field and a magnetic field that are perpendicular to each other, traveling through space. So if a piece of light, a wavelength of light on the top of the particle, the electric field on top would push the electron cloud down, and then when the electric field goes to the bottom of the particle, it would push the electron field up. And so the electric cloud around the particle would go down, up, and it would go back and forth, what we call an oscillation. When something oscillates like that, it is absorbing light. So it can preferentially pick out pieces of light. So white light, you might know, is all the wavelengths, the whole rainbow, all at one time. Gold nanoparticles selectively interact with one part of that light. They reflect the rest, and that's why they look red in color. So something that I've been interested in for a while is, can we use these unique electronic properties of gold to do something else besides just give it color? So they're very nice for chromophores. They're great for making pregnancy tests. They're useful in a number of applications, but can we use gold nanoparticles for other applications, taking advantage of this unique photophysical property? And one thing that I worked on in graduate school was to take a fluorophore. A fluorophore, is an organic molecule that absorbs light, hangs onto it for a little bit, and then releases that light later on. That's called fluorescence. So the difference between that and, say, fluorescence just means something like a glow stick. You crack a glow stick open, something that emits, that's phosphorescence, but something that emits light, not just absorbs it. If we take that fluorophore and put it into a silica nanoparticle, silica is just glass, so little glass marble, we can then decorate the outside of that silica nanoparticle in little receptors and bind to those receptors gold nanoparticles. And because gold has that unique electronic property, it can interact with the fluorophore and make the fluorophore glow brighter. So this was known before, but it turns out that there's a Goldilocks distance. If we get the fluorophore and the gold right next to each other, it turns the fluorophore off. If they get too far away, they don't know anything about each other. If you get them just next to each other, just right, then that little electronic cloud can interact with the fluorophore to make the fluorophore glow brighter. And this is useful for a number of applications. You can think about if you had to have contrast put into your body for medical imaging, you probably want as little contrast as possible put into you. It doesn't belong in there. You need just enough to get the image you want, and then you want no more. So having bright biomarkers is an important aspect that this could help. So that's a graphic of it. This is actually how we did it. I won't go through all of it, but there's some surface chemistry here that's pretty cool, some organic transformations. Turning this group on the end, which is called an amine, into this guy, which is called a dithiocarbamate. And it turns out these sulfurs really like gold. So we can make the silicon nanoparticle really enjoy the gold surface and get these things to aggregate. Turns out they aggregate like gangbusters. So this is a transmission electron microscope image. This is a scanning electron microscope image. And the main difference here is that in one, the electron beam is going to pass through the sample. So it kind of looks like a negative film almost. You see a flat image. And then the other one, the electrons are going to bounce off the surface and come back to the detector. So they will give kind of a three-dimensional look when you aggregate these together. The light gray spheres are the silica. The white balls on the end are gold. And if you overlay a picture of what's called a backscatter image, you can make a more impressive view of this. This is still a scanning electron microscope image. But here you can see the brighter particles reflected electrons better. And so what I'm getting at here is that gold is actually a mirror. When an electron hits it, it bounces off. It bounces back with a lot of energy. The glass absorbs the electrons, and they don't bounce back with as much energy, so they look a little bit darker. So we were able to show definitively that we were able to aggregate these particles, and because of that, we had huge enhancements in the fluorescent brightness here. So our silicon nanoparticles with dye in them looked like this bottom line. So GNP here stands for gold nanoparticle. It is a flat line, no light absorbed without gold present. When we put gold in and changed it, switched over the groups to that dithiocarbamate, the group that likes to bind to gold, we saw a huge enhancement from basically no intensity up to about 700 or so. And as the days progressed on, it actually maxed out our instrument. So we weren't able to tell what the highest enhancement was. So the more and more gold that bound in these particles, the brighter they got. And we were able to show here as well that our surface binding did matter. If we have the particles and the gold nanoparticles together, none of that group that liked them to bind, nothing happened. It looked similar to the silica with no gold present. So this was nice for us because it was a proof of concept that we can organize these nanoparticles, but the amount of aggregation that we got was not easily controlled. And if you ask any scientist anywhere, the thing they want most in their experiments is for them to do the same thing every single time. It is so frustrating to run an experiment and then run it again and get a slightly different result, and then run it again and get a slightly different result. And we said in the paper, hey, we got this amazing result 20% of the time. It was really hard for us to go through and replicate this over and over because we were basically wanting these particles to just mash up together in solution. And that's a challenge to say that they all need to bind this way. Sometimes a bunch of the silicon would bind, sometimes a bunch of the gold would bind. They didn't always mix the way that we wanted them to. And so that has led me to want to explore trying to make more organized structures. And in particular, one that I'm interested in is using gold nanorods instead of gold nanoparticles. So these are unique because nature really likes to make symmetric things. If you think about all the planets, they're spherical. You might take that for granted. None of them are shaped like a donut. None of them are shaped like a rod. There's a reason for that. Raindrops, when they fall, are spherical. They don't fall as little cubes or triangles or pyramids. There's a reason for that. These are low energy structures. Spherical, symmetric structures have the lowest possible energy barriers to form. Trying to tell gold to grow into a pill shape, or we're going to call these rods, even though they look more like capsules, is again what nature wants to do. Naturally, as we're going to add gold atoms around a sphere, it's going to want to turn more and more and more into a sphere. But if we play some tricks on it using a type of chemical called a surfactant, this particular surfactant is called CFAB. It's got a little positive head group on the end, which will be important later on. Is used to make this little cage. It's what's called a myocell. It's just a soap cage, basically think like we're making sausage here. This is the casing that we're going to pack the stuff into, and the casing itself is going to determine the shape of the particles we're making. So using this soap and some silver, we can make this cage, put in some gold into it, and then grow the asymmetric particles. And the real neat thing about these being asymmetric is that instead of them absorbing one type of light, they now absorb two types of light, or what we call in the biz, we have two modes of oscillation. That electronic cloud I talked about can oscillate back and forth across the long axis, what we call the longitudinal axis, or it can oscillate back and forth across the short axis, what we call the transverse axis. And that leads to these two modes of absorption. The transverse almost always sits at this peak here. So it's about 525, no matter how you make the rods, they absorb 525 at the transverse. But as the rods get longer and longer and longer, we can tune that longitudinal peak. So you can see here, up at the top, this is called the aspect ratio. It's a measure of the difference between how wide the rod is and how long the rod is. But basically, the mantra is as the rods get longer, the wavelength gets longer and longer out this way. So we can tune in what sort of light we want the gold nanorods to absorb. My students have gotten very good at this. When I highlight, in particular, Stephen Kane started in my lab. He was one of my first students in my lab and really developed a robust protocol for making these gold nanorods. And we're in the process of putting together a step-by-step guide for how to do this. It's been incredible. When I was trying to make gold nanorods in grad school, I was, again, not very efficient at it. Every now and then I would get the particles. Stephen can make these better than I can at this point. So much of the research all of the research that goes on here is really driven by our undergrads, and so I want to highlight them as I can. Oh, we have someone else that wants to join. Hi there. So you can see here that the gold nanorods grow from taking a seed solution. The seed solution is made up of little spheres of gold. This seed solution is a little older, so you can see it's kind of turning red. The seeds are getting larger and larger, so they look like spheres. When I say seeds, I mean really tiny particles. I know I'm already talking about tiny particles, but really, really nano particles. But when the gold grows, you can see this solution on the side is kind of a light blue color. That is a solution of gold rods, but because that longitudinal axis was tuned to such a way, that blue color is coming from the electrons oscillating back across the long axis, which is pretty cool because we can take gold, which should be golden color, and we can make it any color we want by growing or shrinking the shape of these rods. And I will come back to why that's, oh, click up the screen. Nope, I'm going to talk about it right now. So the reason that this is important to us is that the model I showed you earlier, where I had some fluorophore, some sort of dye that was interacting with gold on the outside of it, is kind of a one-way street. That was the fluorophore, the gold nearby that just made the fluorophore grow brighter. Something that's very interesting to me and to a number of people is seeing if we can use this oscillation of electron density to communicate information across the gold nanorod. So can we take a silicon nanoparticle that has one dye on one side, transfer the energy from that dye into the gold nanorod, and pick up the energy on the other side with a different dye? Biochemists do this all the time. It's a process called forced or resonance energy transfer, or shorthand FRET, where two dyes pass energy next to each other. But they have to be really close for this to happen. What we're interested in is, can we use a gold nanorod kind of like a fiber optic cable? So with a fiber optic cable, you shine in light on one side, light doesn't emit anywhere along the cable, but it comes out on the other side. We want to know if we can do that on a very small scale across a nanometer dimension. There's numerous applications for this, but none are quite as exciting, I think, as for photovoltaic cells. One application, and I've got a used car sales pitch here that I'll briefly throw out because this is basic research. But one of the biggest problems right now with photovoltaic research is that we have two different environments that we're concerned with. One where a photon liberates an electron, and one where an electron then goes on to create a current. Right now, that has to happen in one environment. So a photon hits the surface of a photocell. It needs to liberate an electron and then get that electron to go into a wire. But those two events are not so in agreement with each other. Most of the time what happens is a photon hits the top of the solar cell, the electron is emitted to make what's called an electron hole pair, and they just recombine, which heats up the solar cell, creates no current, and reduces the lifetime of the solar cell. And so if you've seen anyone with photocell or with solar panels in their house, the entire roof is covered in solar panels. And they get maybe enough energy to run a dishwasher. That's because most solar cells right now are between 12 and 20% efficient. So if we could step that up even just a little bit, we're talking about a game changer for renewable energy. One of the potential applications for this system is if we can put an electron liberation event, the photon excitation event in one environment and the electron liberation event in a separate environment, that will allow us to make both those processes potentially more efficient. And that's one area that I'd be interested in. One, seeing if we can get this to work, and then two, trying to put it into what's called, effectively what I'm describing here is called a dye-sensitized solar cell. I'll leave that alone. That is beyond our current research.
Just curious, what sort of energy losses are you expecting from something like this? Or do you know, or do you have?
This should be, so if it works the way that I'm hoping it does, forced or resonance energy transfer is lossless. So it's not the donor dye accepting a photon, emitting a photon, and then this other one catching it. It is an excitation event leads to an emission event on this side.
Very nice.
Which is lossless. Yeah. So that's a good point as well. If we're trying to target one particular wavelength of light, we don't have to worry about losing energy as it's transferred across the barrier. This is the physics for how it should work. So I know there's a lot of lines up here, but the dotted lines mean light that's going to come in. The glowy lines are light that will be coming out. And so the way that I envision this is that I'd use one sort of dye that has an absorption over here around 600 and an emission just a bit beyond that, say 650 or so. That would overlap with the absorption of that longitudinal plasma. So this green dotted line is where the gold sits. The gold absorbs the light from, or absorbs the energy from this dye, what's called the donor dye, which then overlaps with the excitation of the acceptor dye, so that the acceptor dye can then emit. So what I would do is, along this scheme, I would irradiate this sample with 600 nanometer light, and I would expect to see 810 or so coming out on the other side. If it doesn't work, I would irradiate this with 610, and I would see it emit at about 650, because then my donor dye is getting excited and just emitting, it's not transferring its energy. Nope. Okay, so how to do this? When I showed you earlier that I had silicon nanoparticles and gold nanoparticles, it's a crazy mixture of silicon and gold all over the place. I need one particular type of silicon nanoparticle to stick to one side of the gold nanorod and have a different silicon nanoparticle on the other side. Nanoparticles are like misbehaved children. If you put them into a room, they're going to do the exact opposite of what you want them to do. This is what I want to have happen. They're going to be binding all over the place, falling apart, having tantrums. So how do you impart order to this sort of system? And one of the ways that we're attempting to do this is to use DNA nanoparticles. So we have a grand total of 3 nanoparticles you need to keep in your mind. I'm not talking about silica today, but silicon nanoparticles. Gold nanoparticles. Now this is the third one, DNA-based nanoparticles. This is a fairly new field, but one that I think is really, really exciting. It came into being in about 2006, which is bizarre to me because when I show you how it works, it's complicated, but it also is beautifully simple, I think, in the process of how this comes together. What happens is you take a long piece of DNA. In most cases, you use a circular piece of DNA, but it doesn't matter. It can be any shape you want. Let's say this is my piece of DNA. I wanted my piece of DNA to not be flat like this, but I wanted it to curve back on itself to make kind of an S pattern. I could then pick out, this is single-stranded DNA, and most of you may know that DNA is double-stranded, so there's two strands of a biopolymer put them together. I would take my single-stranded piece of DNA, and I would put in complementary strands that bind across these gaps to hold it into this shape. So then I can use what's called the Watson-Crick base pairing to make these shapes. The DNA builds itself, what's called bottom-up construction, and put in all the pieces that I need. I map it out on paper and say I want it to make a certain shape. So I take a strand and I raster, or if you think of an Etch-a-Sketch, if you ever drew anything with Etch-a-Sketch, it's called rastering, where you take a line and draw something back and forth with it. I take a line here and I raster it back and forth, to make a square. And then I put in strands to hold it in that square shape. This is a model of what it should look like. And this is something called an atomic force microscope. I'll come back to how that works in a second. Image of it. And I can do this with all sorts of shapes. So here I made a square that was just all solid down the middle. I had a square with a seam down the middle, stars, smiley faces. This is real data. So the little smiley face over here, that is DNA. And you can see, I know it's a little small in the corner there, but there's billions of them. They actually took a video of these when they were depositing them, and there's little smiley faces just floating around the sample. It's pretty hilarious, but it's more impressive when you think about the width of this smiley face is 50 nanometers. I didn't make a big stink about this at the beginning, but when we talk about nanotechnology, it's important to kind of take a grasp at how small we're getting at here. So a popular example is, if you think of a penny, an older penny, because the new pennies are different, but an old penny on the back is the Lincoln Memorial. If you envision an eyelash on Abraham Lincoln's statue in the Lincoln Memorial, that eyelash would be 100 nanometers long, roughly speaking. So we're talking about very, very small. So this, we could fit a couple of these on Abe Lincoln's eyelash in the back of the penny, but there's such fine detail to it. And that's really the exciting part here. We can make cute shapes with them, but what I'm getting at is we have fine-tuned control over these structures at a very, very unprecedentedly small scale, which allows us opportunities to organize at that scale for the first time. This is a video of how these come together. I didn't write this up. Unfortunately, the author, a professor from UC San Francisco, didn't put his credentials on this. This is not mine, but it was hard to cite on top of the video. The gray strand that you see coming in the top is what's the scaffold strand. And all these little colored strands are staple strands. They're the strands that are going to hold the scaffold strand together. The scaffold strand would be some natural source. We use like a bacteriophage genome or something like that. But all the little colored strands are synthetic. So I can call up a company and say, all right, guys, I want you to send me a strand that says A, A, T, C, G, so on and so forth. And I would know where each of those strands are supposed to go. Play it one more time because I think it's a pretty cool video. I would know A's bind to T's and C's bind to G's. I would know which strand is going to bind to which part. And so I would map out where I need each one of these strands to go. And then my job is simply to just mix about, in my case, 200 staple strands with one scaffold strand, heat them up, and then very slowly cool them down and let DNA do its job. The A's find the T's, the C's find the G's, and they make the structure that I told it to make. So I could even draw this out on paper. We've got some fine computer programs to do this for us, but you can actually do it on paper. It works out reasonably well. The other thing that's really exciting about this is that everywhere that I have put in a staple strand, so this is a map of a construct that we use. Everyone can see all the details here? This is, no. I'll come back to this structure here, but these two big planks, what I've done is just taken a snippet on one of these planks. So this is what it looks like on a zoomed in level. The black line and the black letters represent the scaffold strand. So it's going to just raster back and forth here. And then all the colored strands here are staple strands. So this is a bacteriophage, and I'm sorry, a bacteriophage is a virus that preferentially affects bacteria. So we're taking a viral gene. Viruses like to use single-stranded DNA, and inject that DNA into us, bake us into using their DNA to make their proteins. That's what it gets their meat. But we get back at them because we're using their genome to reproduce. So these staple strands are synthetic. So I can call up my company and say, I want it to say TGAATCTT, But then I can do something else that's really unique. At that T, that particular T, with sub-nanometer precision, I can say, I want that T to have a tag on it. So I want it to still be a piece of DNA, but I want a tag coming off of that, and I want to put a sulfur at the end of that tag. Remember, I brought up earlier, sulfur and gold really like each other. So what I can do is put a little marker here at that T that says gold should bind to this particular part of the DNA origami, because gold and sulfur get along. Now, one sulfur is not enough, so then I go over to this A, and I say, well, that A is pretty close. Let's put another sulfur on that group, and I can decorate this whole area where there are staple strands with sulfurs to bind an object specifically at that point. Real quick, because I'm going to bring up some of these about how these are imaged. I mentioned atomic force spectroscopy. We have to get really creative when we're trying to measure these things. We're smaller than wavelength of light, so we typically use an electron microscope instead of an optical microscope. But with DNA, it is paper thin. It's really small. And so it's too small to stop an electron beam. And I talked about gold nanoparticles and silicon nanoparticles. They have kind of some three-dimensional shape to them, so they can slow down the beam. But origami is really bad at this. DNA is really bad at this. So instead what we do is we kind of feel the surface with a little tip. And this works almost exactly like how a record player works. So back in the day, we all used to listen to music on things called vinyls. I don't need to say that in here for most of us, a couple of us. Right? But vinyl records worked by having a series of bumps on a vinyl surface. We put a needle on that surface, the needle would bounce up and down, and based on the number of vibrations in that particular area, it would play music. The same thing is going to happen here, except instead of having music play, I have a series of DNA structures that are deposited on the surface, and I just run a tip over it. So the tip says, this is flat, oh wait, something's tall here, this is flat, and it moves up a row and scan, moves up a row and scan, and like that over and over and over. We get this raster back and forth, back and forth. Well, this is pretty neat because it tells, if I know exactly where that tip is, I know a X, Y dimension, but I also know height, so I get Z dimension from that as well. And we can produce three-dimensional objects by scanning along the plane, but also up and down. So this is kind of a three-dimensional representation where you can take that object and turn it sideways. We know that the tip is bouncing up and down based on a very fine-tuned laser. So the laser bounces up one side and hits a diode over here. So as this tip bounces up and down, the laser moves around. So I'm going to show you a series of atomic force microscope images. It's basically just a record player imaging the surface of these DNA structures that are attached. One other thing I want to bring up before I show you the full structure here is that this diagram confuses a lot of people when I show it because each one of these planks I want to represent by these little graphics here. So you might notice there's a hole in the middle. So I'm going to represent that by just a rectangle with this white square. What happens is that the plank on the right overlays with the plank on the left. So we're going to pick this one up, turn it 90 degrees, and they're going to sit on top of each other. What that's going to do is create this little plus sign, but because there was a hole in the middle of this one, there's an area where these overlap where the DNA is twice as thick. There's basically two pieces of paper laying on top of each other, so I have a single thickness of DNA, a double thickness of DNA, a single thickness of DNA. Remember, our AFM is sensitive to height, so when we measure these things, we can see that, and it shows up as an equal sign. So this is our DNA construct that we work with, and you can see those two planks laying on top of each other, and you can see the fine detail, all these little equal signs, that is an area that is twice as thick. So our Z-scale in this image is shown by color. Things that are lower or darker in color, things that are closer to you are lighter in color. And this is a blow-up of one of those. You shouldn't have to squint too much. If that looks a little bit like corrugation for you, like expanded steel, what you're looking at is the double helical nature of the DNA. This is a very, very fine-tuned image. And you can see how well defined each one of these aspects are. Again, that's exciting for us because one, can make a bunch of them. You can see I didn't make just one, just a couple around. But they're also very well defined, and so that allows us, again, to impart this organization on the materials. This is an example of how materials can be organized on the surface. So in your biochemistry classes, you may have heard about biotin and streptavidin. Biotin is a small molecule, streptavidin is a protein. And biotin and streptavidin have this big old love affair. They bind to each other with some of the highest affinity that we've seen for any biomolecule. So they make popular tags because streptavidin really likes to bind to biotin and not let go. We showed that you can label the edges of our DNA origami, of our plus sign, with biotins. So this is a small molecule, and I've told them where there were staples, I said put a biotin there and there, and a biotin there and there. And then we can mix those with streptavidin, which is a protein, so it's a little glob. And when you mix those together, you can see it's a little harder to see in this image, but in the blow up, you can see a dot there and a dot there. Those are the streptavidins that have been bound to the outside of the origami surface. So the proteins went exactly where I wanted them to go. So basically saying that I've organized my proteins on the outside here. And now what I want to do is try and bind things other than just proteins, namely gold and silicon nanoparticles. So that brings me back to this thing that I brought up earlier. I want to make an object where I can transfer energy, but I need to organize them like that. Let's stop using the little kid graphic and talk about this more in terms of how this is going to look for research that's ongoing currently. This is obviously the stylized version, but I think pretty slick graphic showing a didoped silicon nanoparticle next to a gold nanorod, next to another didoped silicon nanoparticle. You might notice this one is blue, that one is red. And then underneath here, a really big blow up of what would be DNA origami. And the idea is to use the DNA origami as a scaffold to organize these materials in place, and then we're going to use some surface chemistry to actually lock them in place. DNA is not meant to be a structural unit. So if we rely on DNA holding these particles, together, this probably works. If we rely on DNA holding these particles together, then I've made kind of a bad material. Because if it gets too hot, if the salt concentration changes, if it gets too cold, if someone doesn't look at it right, the DNA is going to fall apart. Right? My molecular biologist and biochemical colleagues like to in the test, DNA is very sensitive. It is not a very good structural unit. But what we can do is use it to organize materials in advance and then do some chemistry on the outside. I want to focus just on the organizational part. There's another aspect of this that I won't go into today about how I plan on chemically welding these together. But for any of you that are nervous about me using DNA to hold nanoparticles together, if you're not, I have a plan for that. So I'm going to focus just on this aspect of it, of how do I get a silicon nanoparticle to bind here and a piece of DNA to bind, or a piece of gold to bind here. And the way to do it is to use docking and landing strands. So the plan is to DNA coat gold nanorods with a particular strand, and then have the complementary strand be attached to the DNA origami. So I can go to one of the places on my, I'm not going to use the plus sign, but I could use the plus sign here just for fun, and tell it to have a 20-mer single-stranded piece of DNA that is red, and then I would put in the complementary DNA piece on top of the gold. So A's and T's bind to each other. So I could have the piece from the bottom say A, and I could have the piece from the gold say T. They would bind to each other and selectively put the gold at this position. And then I could have C on this part, and G up here to bind the silicon on the other part. So the blue strands of the silica would be different from the red strands, and they would not want to bind to each other, but they would want to bind complementary pairs on the origami. So putting DNA on DNA, not a problem. It's very easy to go in and say, I want a strand of DNA, but I also want another single strand coming off it, so like a T-stranded piece of DNA. That I can order, it's commercially available, and wouldn't you believe it, it's super cheap to do this. So back in the day, it was quite expensive, but you can go to a company and they have a little chat box, and you type in ATCG, literally use a keyboard, type it in, you push send, and 2 days later, someone sends you a vial with a piece of DNA in it. It's amazing. It costs like $30 to do it. It's pretty cool. So I'm not so worried about that. The part that we are working on currently is making gold nanorods and decorating the outside with DNA. So I want to talk about some of the advancements we've made in that area. I showed you the student who came up with our protocol for making gold nanorods. I also want to highlight Michael Smith, him up in the corner over here. Michael did a summer research project through INBRI, that's through the National Institutes of Health, to DNA decorate these gold nanorods. So over the course of the summer, he was able to make a lot of headroom, a lot of progress in this project. We don't need to go into all the details here, but basically what he did was he took a piece of, this is our piece of DNA, so it's a little more complicated than AAAA, but it basically has a common strand to it. And then on the outside, there's this group that has sulfur on it. It's in a type of packaging that doesn't like to bind to holes. What we have to do is turn that sulfur into what's called a thiol. We need to break this bond. This particular type of bond is called a disulfide. Disulfide bonds show up a lot in nature, but they don't bind to gold very well. So we need to break that using this molecule called TSEP. I'm only telling you these details because they're going to show up here in a second. This TSEP part will come back. But when I turn that disulfide into this thiol, now that thing really likes gold. So I can mix in my gold nanorods after treating them running through a number of processes and hopefully coat the outside of the gold nanorods in this DNA, from these DNA strands. A couple things need to happen when that occurs. We need to have a couple of checks along the way. These are the DNA. These are the gold nanoparticles before they have had DNA coated on the outside. So they still have that soap coating, the thing called CTAB that I brought up earlier. These are the DNA or the gold nanoparticles once DNA has been added. So you can see they still look like DNA, or they still look like gold. And it might be clear or not, but there's actually this little shadow on the outside. The DNA is not long enough that we should be able to see it, but it looks like when you DNA coat, there's a certain amount of salt aggregation that comes up as well. And so you can almost see kind of like a halo sort of structure or an outline around each one of these gold nanoparticles. That's not enough for us to say it's DNA coated. I've got a better description of that in a second, but it is convincing at least to say that the gold nanoparticles are still particles. They haven't turned into something crazy. We can use our light measurements as well to see this. So you can see here when the gold nanorods are CTAB coated, that's the blue line, looks like that. When we wash them, so that is we remove some of the excess soap, the CTAB, the soap that we work with, doesn't like DNA very much. It's kind of why we wash our hands. It's really good about removing biomolecules. So we want to put a biomolecule in there and get rid of the soap. So when we wash them, the peaks change a little bit, but not much. When we put DNA on there, again, the peaks change, but not very much. So you can see that what we're doing is just exchanging one coating for another coating, and the gold nanorods are playing along each time. That's important to us because we need that property, we need the way that this interacts with light to maintain itself as we go through all these recoatings. If we recoated the gold in DNA and it totally lost its physical properties, they're no longer of interest to us. They need to have both. So this looked promising. The results that we got back from the TEM looked good, but this is not convincing enough to say, are they actually DNA coded? I don't know that this particle doesn't have DNA and that particle does. It's too small to see in an electron microscope. So what we did is used a gel electrophoresis method, which is commonly used to separate out proteins and DNA molecules based on their charge and their size. So what gel electrophoresis is a maze of a polymer called agarose that creates a gauntlet for DNA to have to run through. What you do is you take, so just to lay us out here, this top image and this bottom left image are the same. It's just two different ways of looking at it. This is under room light, and this is looking through UV light. So I'll come back to that. But under the room light, so you can see all these, you'll see a series of squares up here. Those are wells that you can put a little bit of sample into in solution. And then this material that looks kind of cloudy is agarose that creates the maze for us. You put one of these into a gel electrophoresis box, and you apply a positive charge up here and a negative charge down here. DNA, by its nature, has kind of a negative, opposite way, negative charge up here, positive charge down here. DNA, due to its nature, has a slight negative charge to it. So when it comes near a positive charge, it will run towards that positive charge. So we can use basically put a plus sign down here and pull, when I turn the field on, pull the DNA towards the plus. Because it has to run through this maze, it will separate out based on its size. Really small pieces can wind through the maze quickly, and they run down the gel fast. Larger pieces can't run through the maze as quickly. Okay, so that's gel electrophoresis. The reason that we used it here is to show that we wanted to show that we actually had DNA-coated gold nanorods. So from left to right, the way that we did that, with very much thanks to the DOTS Purdue lab, was to use a series of controls to say that, are we seeing DNA, are we seeing gold, are we seeing both? Because there's a number of things in these solutions. On the left, this lane contains something called a ladder. It's got several pieces of DNA that we know what each size is. So there's very small pieces up to very large pieces. The small pieces should run down the lane very quickly. The large pieces should get held up. So that allows us, if we were trying to guess what the size of the DNA is, we could use that ladder to show. In our case, we're not so interested in determining what the size of the DNA is. We know what it is because we've ordered that DNA, but it can be helpful. The next box over here are 3 oligonucleotides. Oligonucleotide is basically the same DNA, so that's just the DNA that's not been bound to anything. And so you can see here that this band shows up. because we stained it with this particular substance called potassium bromide, it lights up when it interacts with DNA. Turns out it's not very healthy to hang out with, though. Molecules that really like DNA, typically not very good for us. If something likes our DNA, it's going to mess with our DNA, and that's going to get cancer. So we have to be very careful when we're working with this potassium bromide. That's what this ETBR stands for. So you can see when we don't have when we have just DNA put into these wells, it runs down to here. And then these next two lanes are gold nanorods that have DNA attached to them. You can see those same bands here. This is likely the free DNA. But what you can't see is the gold. If you go up here, remember those samples that I showed you where they were kind of blue? You'll see a light haze of that blue. That is the gold nanorods that have been pulled through this gel. Now they lag behind the free DNA because they take up more space than the DNA does. The DNA is going to run ahead of it. You can see the stain that we put in here. The DNA will be just ahead of it. This is just the stain. But you can see, I'm sorry, the DNA will be just ahead on this side. You can see the gold nanorods travel towards the positive terminal. That is different, and these are just two trials that Michael tried here. That is different than when we have, well, the washed particles we'll have to come back to here, but the washed particles are a little bit of a conundrum, but when we look at the unwashed gold nanoparticles, CTAB and TSEP, so in particular, please cut off. Washed and unwashed, I'm sorry, that's these two lanes. We have a lane that mixed up in the middle. So when the gold nanoparticles don't have DNA coated on the outside, the nanoparticles will run opposite because they're coated in that surfactant that has a positive charge. The positive charge is going to be attracted to the negative terminal, so we see them run in the opposite direction. You can see that's also true when it's just CTAB in solution. And I'm sorry these got mislabeled. Let me get that fixed for the next part here. I borrowed these from one of our posters. But we were also able to show that TSEP here on the outside doesn't play a part in this. When we put that TSEP molecule that broke up the disulfide, it shows running in the normal direction as well. So what that tells us is that when we have CTAB coated gold nanoparticles, they run opposite, as we'd expect. They have a positive coating on the outside. When we have DNA coated gold nanoparticles, they run towards the positive terminal. And so this was a definitive experiment showing that we indeed have DNA coated nanoparticles. And this is very helpful because now we have the gold all in place. And so we can turn our attention now to DNA labeling silicon nanoparticles as we still build up our DNA origami structures. There's quite a bit of work still ongoing here, but I wanted to highlight some of our recent successes. This was very exciting for us to be able to show this using gel electrophoresis instead of just having our normal images. These are the links to keep it in focus. I didn't go through all these. So my hope here is that, one, I didn't bore you all to death, in that I was able to impress upon you that gold nanoparticles are pretty interesting. You use them more than what you probably think that you do, but they're better for, they're useful for more than just colors, that hopefully we can aggregate them in useful ways to get bulk properties out of them from nanoscale. And none of this work would have been possible if it wasn't for all of the people that work in my lab. I've maintained a rather large undergraduate lab, and I rely on those undergraduate students to really be rather independent. So they come into lab, we go through safety and through some of the basic experiments, but then I have to rely on them to become rather independent rather quickly. Running primarily undergraduate research is a challenge that I think really benefits our undergraduates because they have ownership of these projects. So I highlighted in particular Stephen and Michael for these two areas that I was talking about, but there's a number of advancements that we've made with undergraduates who, if I was half as talented as they were when I was an undergraduate, I'd be a Nobel laureate right now. So we've got some really amazing students. This is my current group. We've had a series of students go on to be very successful. None have gone on to graduate school just yet. I haven't had any converts, but I'm working on it. But here's some of our initial group, and then this is our group at the latest American Chemical Society meeting. So I'm very grateful to all of them. I'm also very grateful to NSU, to the Faculty Research Council for providing startup funds for this research and continued support from the NIH in putting money towards this. I'd be happy to answer any questions that you might have.
So your next step is to see if you can coat the silicon for the other end of the particle, is that right?
That's right. So we've already, I didn't show it, but we have already made the di-dope silicon nanoparticles. So now we're interested in doing surface chemistry on the silica. So we'll do a couple similar surface attachments and similar gel electrophoresis experiments to show that they are DNA coded. And then when we have those two particles in hand, we'll get to the work of building the DNA scaffold that we're going to use. The plus sign that I showed you is useful for a couple applications, but we're going to try and build something that's a little more rigid. And molecular models of that cross-shape origami I showed you, they're kind of floppy. So we need to build something that's a little more hardy, like an actual piece of scaffolding. And there's some good precedent for how to do that.
So I have to ask, how big is the free oligonucleotide?
20 base pairs.
Why do you see two bands on your gel?
That is an excellent question. I think that what we took, so what Dr. Dosperdu is asking about is these two bands that show up.
Yeah.
I don't know if it's a consequence of how short we ran it because of the loading die, if that normally would run off, and that we were running it for such a short amount of time with such a high percentage agarose. So we ran these, I believe these are, I would say 2%. And so maybe there was part there, but I don't have a good explanation for that, why we see two bands.
Why is trial 1 and 2, why are they look different size?
What are they different? Oh, there was almost certainly a different concentration between the two. So this one looks a little bit lighter.
Fine on that gel, but the UV, why do they look like as if they're two different sizes? They should be the same, right?
That's right. They should be running about the same size. I don't know if it didn't run evenly or it could be. So this lane had obviously a higher concentration of gold nanorods. I don't know if that drug back the DNA as it was trying to run through.
Yeah.
Because it could be that there's some light association between these, but those are the particles, or those are the oligonucleotides that weren't fully bound to the gold surface. I can envision maybe a late liberation of DNA here compared to earlier liberation in the others, or non-binding altogether.
So these are completely two different trials.
That's correct. Yeah. We put them onto one gel. Typically, you would not load them onto one gel, but because we had the generous help of you and your students, we wanted to make sure we asked as little of the lab as we needed to, so we put them on the one. Dr. Paulson Cambiano.
Can I go ahead? Can you hear me?
Oh, yeah, and there might have been another one here, but go ahead.
Can you hear me okay, Nathan?
I can.
I get the impression that you're using very short sections of DNA to coat your nanoparticles, yes?
Yeah, they are 20 nucleotides long.
What do you think is the limit for how big a section of DNA you could put onto a DNA, to a gold nanoparticle, or do you have any idea how big a one you could get?
Yeah, you can put very large, so as long as you want. We intentionally stay away from longer oligonucleotides because you have non-specific binding. So what that means is that they will, if you put a long enough section, think about if I have a dictionary's worth of words, some words in that dictionary are going to line up with another book. I want a very specific binding. And so with shorter oligonucleotides, you have less non-specific binding. They only bind where I tell them to bind. And importantly, when they bind inappropriately, they don't bind very well, so they break apart. If I used 100 MERS and 20% of the strand stuck, that might be enough for it to bind in the wrong spot. And so we try and intentionally stay away from longer oligonucleotides.
So what's the minimum?
That's a good question. I don't have an answer for the minimum. I would say, based on simulations that I've seen, something called toe hole strands, that is where you want a strand to invade another strand, tends to be 3 to 4 nucleotides long. Basically, if I had two pieces of DNA stuck together, and I was going to show you a video of them, the ends would be flopping around like this. The middle would be bound together pretty well because it'd be protected, but these would be peeling back and binding over and over in solution. And so if I'm wanting one thing to be down here and one thing to bind to it, I'd probably want them to be longer than, say, 3 or 4 nucleotides. But it may be possible. You can envision trying to get those to stick together. You would just need colder temperatures. And the warmer it is, the more binding and unbinding you're going to have between those two. These are hydrogen bonds holding these together, so it's not a massive amount of energy.
When you're doing the gold nanorods and you need a specific length for a specific wavelength,
Mhm.
how do you, is it the amount of time that you let them in solution that?
They are really, really picky. It's not the amount of time. It is a combination of the amount of silver. So there's a little bit of silver that we put in there, and it's a number of things. It's the relative amount of reducing agent, so turning what we use to turn the gold 3 into gold 0, the amount of gold that's present in the solution, the amount of silver that's present in the solution, and the amount of soap, that surfactant that's in solution. You can dial in each one of those things and say, if I add X amount of silver, I'll get this rod. If I add 2X amount of silver, I'll get this different rod. But the temperature matters. If it's sunny or cloudy outside, it matters. And so that's part of the, I'm kidding about the sunny and cloudy, but they're very sensitive. They're very particular about being formed. But because we put these things into solution, they will run overnight. The reaction will be complete overnight. And whatever conditions we've given it, that's the rod that we grow.
It's a well-known process and repeatable.
It is a well-known process. It takes expert hands to put it together. It's not something, if we brought in a bunch of elementary school kids, I'd say, all right, kids, let's all make gold nanorods, because most of the class is going to be upset at the end of that. So. If no further questions, thank you all for your time. It was fun to chat about nanoparticles. If you're interested in any of this work, come on down to the lab. We love having visitors. And to Mr. Gore, we're always looking for talented new students. Or yourself, sorry, I don't know you, but
Jasmine's husband.
oh, well, if you want to do some research too, Jasmine's husband, then come on down. Thank you, guys.
The Faculty Council would like to thank you all for joining us in this Hawk Talk event. We will have 2 additional Hawk Talk presentations in the coming months. Our next Hawk Talk event is slated for October 30th, and that's going to be at noon. We'll have Dr. Sanjari Ghosh. She was here a little earlier, zooming in. She will be talking about the economics of water trading in the Western United States. And then on November 27th, we will go back to our 3 to 4 time, and we'll have Dr. Mia Revels talk to us about northern saw-whet owls in Oklahoma, her research on that. So check your calendars, and we hope to see you at our future Faculty Council Hawk Talk presentations. Again, thank you for joining us today.
Thanks.
Thank you. Thank you, Pam.
Thank you.
So to click, what's the wrong thing?